Peter White's here with In Touch in 40 Minutes. First on BBC Radio 4, 10 years after the emergence and worldwide spread of the disease SARS, Alok Jha reports on efforts today to predict and be better prepared for future pandemics of deadly infectious diseases.
The World Health Organization says the number of people in China who've caught the flu-like illness, severe acute respiratory syndrome, is rising rapidly. More than 130...
We had no idea what it was. We had no idea how it went from me to you.
Hong Kong's health system has been thrown into crisis by the spread of the SARS virus.
Not only were patients coming in who were very, very sick and dying, but nurses, doctors were catching the disease and dying, and that is terrifying.
The number of people around the world killed by the virus SARS has risen to 98.
The World Health Organization issued a travel advisory this morning that recommended postponing all but essential travel to Toronto.
So far, nearly 200 people have died from severe...
If you have been exposed to SARS, stay in quarantine.
It caused fear around the world, cost billions of dollars to the global economy, killed almost 800 people and infected 10 times that number. A decade ago, an outbreak of a new virus, SARS, had everyone on edge. It could have been worse. The Spanish flu outbreak of 1918 killed an estimated 50 million. And that was in an age when the fastest way to move between continents was by ship. In today's airplane era, a new pandemic-causing virus could emerge in Beijing and arrive in London within hours. The most pressing threats come from zoonotic viruses. These are viruses that jump the species barrier from animals and into us. SARS originated in bats, Spanish flu from wild birds, HIV from Central African primates. The list goes on. In this programme, I'll be asking if, assuming a future pandemic is inevitable, can we predict it? More important, can we do anything to prevent it?
There are numerous zoonotic animal pathogens out there that might cause pandemics in the future, and we know that they may because they've done it in the past.
One of the reasons why this is such a dangerous thing is because it's a new disease in that new species, and so firstly, we won't know what it is when it first happens. Secondly, we don't know how it's going to affect that new species.
We do not understand what it is that drives viruses to jump from an animal into a human population, and what it is that drives, once it's in me, what it drives the ability to go from me to you, human to human transmission. Those are the key steps, and we don't understand those.
The viruses that most worry scientists have high fatality rates. For example, the avian flu virus H5N1 has killed more than 50% of the people known to be infected with it. But simply infecting a person isn't enough to cause a worldwide problem. To kick off a pandemic, a virus also needs the ability to easily transfer between people. That hasn't happened in the case of H5N1, yet. I've been talking to leading researchers in the diseases that transfer between species to find out what they're worried about. And I've been investigating some of the things they're doing to help us predict and prevent the next pandemic. Viruses from animals are always knocking on human doors. Mark Woolhouse, Professor of Infectious Disease Epidemiology at the University of Edinburgh.
The human population is constantly bombarded by viruses that are essentially trying to get in. The term that people use to describe that process is chatter, the constant bombardment of the human population with potentially new viruses, some of which can get in, some of which can cause an infection, some of which can spread a little bit, but very, very rarely do they actually take off.
That the chatter is usually benign is reassuring, until you think about the consequences of some of those rare events.
We have, historically, the emergence of HIV-1, which of course has caused a catastrophic pandemic over many decades, and that emerged from chimpanzees in Central Africa, and we have an extremely detailed picture of how that process happened about a century ago. There are a number of different lineages of HIV-1 viruses. It crossed over into humans many, many times. But there was only one lineage that took off. There was only one that caused the catastrophic pandemic that we're experiencing now. So even out of the diversity of the ancestors of HIV-1, some took off and some didn't. It's a very complex situation.
When an animal virus first infects a human, the event is called a spillover. These events are happening more frequently now than at any time in the past. Working out how common they are is part of the job of Derek Smith. He's director of the World Health Organization's Centre for Modelling and Control of Emerging Infectious Diseases at the University of Cambridge.
The truth is, we really don't know how much of this happens. Much more is noticed today than was noticed 50 years ago and was noticed 50 years before that. There are reasons to think that this might be because we disrupt habitats, we come into contact with some animals that we haven't been in contact with before, things like this. But we also look harder.
And given the rate people are encroaching into previously pristine environments, we need to look harder, says Jeremy Farrer, director of the Wellcome Trust and, until recently, head of the Oxford University Clinical Research Unit in Vietnam.
As the environment changes, whether that's land use change, growth of populations, movement of people from rural into urban communities, but still keeping their close links to rural communities, so often having relatives, they often go back and forth between these two. In many parts of the world, although living in big cities, they still keep their ducks and chickens and their whatever else in their home environment in the big city. So this change in environmental use and human environmental factors, this is going to drive health over the next 50 years. And if we don't build a proper human-animal health policy framework and practice, then we put ourselves at huge risk of these emerging events.
Fortunately, the race to detect and identify emerging viruses has been helped by a revolution in genetic technology.
One of the enormous technological advances in recent years is the capacity for what we call high-throughput sequencing. So for looking at the genomes, not just of a single virus, but of the entire virus present in not even just a person, but a group of people, a set of samples. And we can screen that to see if there are unusual genomes present very, very quickly.
Take the recent virus that's jumped from animals to people in the Middle East. Scientists discovered very fast that MERS, as it's sometimes called, is related to SARS. Ron Fouchier, a professor of virology at the Erasmus Medical Center in the Netherlands.
In 2003, with SARS, it took us months to figure out what was causing this disease. What the genetic code was even took us 2 or 3 weeks to figure that out. With MERS, we got sent a sample by our friends in Saudi Arabia, and within a day we had the full genome sequenced, and we knew what we were dealing with. So we moved from 4, 5, 6 weeks to 1 day in identifying previously unknown pathogen.
This new generation of virus-detecting technology is being put to work at the emerging disease front line in places such as Vietnam. Ho Chi Minh City, in the south of the country, is the headquarters of the Wellcome Trust Vietnam Initiative on Zoonotic Infections, VISIONS. It's a collaborative effort between Vietnamese scientists, government health agencies, and the Oxford University Clinical Research Unit. They're using the latest techniques to carry out surveillance of humans and animal populations in a number of provinces around the country. Vietnam has been hit by several zoonotic pandemics and outbreaks in the last decade. SARS in 2003, H5N1 avian flu in 2004, and again in 2005. Identifying a new virus as soon as it enters people is the first line of defence. That's the aim of one arm of the VISIONS project. It's working with hospitals around Vietnam to collect and catalogue the viruses that are out there, making people sick. And that includes previously unknown pathogens that might have spilt over from animals. At the main hospital in Dong Thap province, the initiative's principal investigator, Stephen Baker, took me through what his team were doing.
Studying one of the busy outpatient departments at Dong Thap Provincial Hospital in Cao Lan City, located in the Mekong Delta in the southern part of Vietnam. This is a 400-bed hospital performing general medicine for the population of Dong Thap province.
So what kind of symptoms would make a patient here interesting to you at VISIONS?
The 4 syndromes that we're interested in are patients with acute respiratory infections, acute diarrhea, jaundice, and also central nervous system infections.
And so what happens when one of the doctors identifies something like that?
So once the doctor identifies one of those patients, they will be enrolled after they provided informed consent for them to take part in the study. The doctor or the nurse will ask them a range of questions of data that will be associated with the sample, and then they will take the specimens, so then we can perform some analysis here through the existing hospital capacity they have for diagnosing disease, and then those samples will be frozen, and then in batches they'll be shipped back to our laboratories for more in-depth analysis in Ho Chi Minh City. At the moment we have 7 hospitals that are participating, and we hope there to be 8. All in all, we're trying to enrol over 12,000 patients over the next 3 years.
Around the world, a lot of patients come in with infectious diseases, with fevers and so on, that are never formally diagnosed. And these are entered into the medical records of disease of unknown origin. So we can go into hospitals and clinics, often in quite remote areas, and look at cases that people haven't been able to diagnose. See if we are able to diagnose them through the diagnostic tools that we have to detect known diseases, and see if that's what these people are suffering from. But if they're not, there's the obvious potential that they're suffering from something different, something new. So that source of these diseases of unknown origin is a pool in which we can look to see if there are actually other kinds of virus or other kinds of pathogens circulating that we weren't previously aware of.
The most surprising thing to me when we started doing this project is how little capacity there is for actually diagnosing disease in the first place within countries like Vietnam. So I think that in the future, the way I see this type of research going for us in the next 5 to 10 years, we actually have some form of disease surveillance that's set up within the provincial hospitals where they're actually doing and reporting their own strange viruses that they're detecting in samples.
Another side of the Visions project takes the surveillance of viruses to places where people are in frequent and close contact with animals.
We do this with what we call high-risk cohorts. People who work in farms, people who work in wildlife farms, people who work in the slaughterhouses round the back of a livestock market, and so on, who are constantly exposed to whatever viruses are present in the animal populations they're working with.
On the outskirts of Kowloon City, Stephen Baker took me to one of the project's long-term surveillance sites. This small, ramshackle farm is typical of many in Vietnam, with a few pigsties and chickens, some in cages, some running around. And it's not just the human inhabitants that the Visions team is here to sample.
In amongst all these fighting cockerels, which you can hear, there's a very incongruous sight. There's 4 people in what look like hazard suits with a bunch of syringes. What are they doing?
So these are some employees from the Sub-Department of Animal Health who are our collaborators on this project. So what you're seeing is they're wearing biological safety suits to prevent them obviously breathing in large particles of feathers and various other things when the animals are moving around. And you can also see that they have a number of different syringes and swabs and also some vessels for storing the samples in. So they're taking blood samples, they're taking feathers, and they're taking cloacal swabs and oral swabs, which will then be sent back to Ho Chi Minh City where we will process them.
Are you suspecting this farm of anything in particular, or is it just a screening?
No, this is just a screening. Over the next 3 years we'll come to this farm and sample humans and animals at the same time point.
And so the farmers and the family here will also be surveyed to look at whether any viruses or bacteria are transferring?
That's correct, yes. So they've been approached by our collaborators in the province and they've been invited to be enrolled in the study. And then routinely, every 6 or 12 months, depending on the farm or the type of individual, we'll visit this location and we'll take animal samples and we'll also take samples from the individuals that have been enrolled.
And so if there is a virus transfer, it's one of these sorts of samples that will give you the first hints?
That's the concept. The most important part I should note about this study is that any time any of these cohort members get sick, they will contact our representatives in this province, and then we will send healthcare workers over to address them with a questionnaire, and we will take samples from them, and we will also give them some medical advice.
And what's the protocol if you do find something interesting or new in the samples you look back in your lab?
So the protocol would be that we would report anything back to both the Sub-Department of Animal Health and the Department for Preventative Medicine to be used for future investigations or as a warning system potentially for viruses that may be circulating.
Visions also monitors pathogens circulating at markets, where a variety of animals, both dead and alive, are on sale. The project's Mary Chambers showed me around a bustling market with stall after stall of pig carcasses, heads and offal. After walking past all that, we headed into a section selling live animals. As well as poultry, there were creatures caught in the wild here: snakes, frogs and rodents.
There's a lady selling rats here, rubbing the top of the cage to show how alive and vital they are, I'm assuming. There's about 30 or 40 rats squashed into it with a big block of ice to lick, a potato to eat. They're quite healthy looking things, to be honest.
They're field rats, so they'll catch them in the rice fields. They put traps out. They learn where the little rat path is, and then they'll put traps. So there's whole families who live on this industry of catching rats and selling them.
So someone's here to buy a rat. They're taken out, smashed against a rock to stun them, and immediately their heads are cut off. A pair of scissors, legs, head, and tail all chopped up in a pair of scissors, and they're skinned within seconds in a bag, fresh as you like.
It's quite a strong smell of rat weed when you get close to them. It's not an environment I'd like to sit in all day.
At least she's wearing gloves.
Field rat or rice field rat is a bit of a delicacy in this part of Vietnam, and you can see them being sold and butchered for consumption at the market. Rats circulate freely within the environment. They transfer between farms, and the potential here, obviously, as you've seen in the market this morning, the way that they're actually dealt with and butchered at the market means there may be a potential exposure risk for humans in that context also. And we have found that they do carry a number of different types of both bacterial and viral pathogens. So the potential risk for infection is there. Whether they're going to be the source of the next pandemic, then we're not so sure.
It's important to survey wildlife because so many viruses originate from wild species.
There's a huge interest in wildlife, and there's many different groups that are interested in sampling wildlife for these potential zoonotic exposures. We are working in other provinces that have more wildlife and more frequent wildlife that we can access, and we're sampling civets, which are large wild cats, bats, but also farmed porcupines and other things that occur in other provinces. So there's a combination of wildlife that's farmed here and wildlife that is actually wild.
You heard Stephen Baker mention bats there. Of all the wild animal species, bats have grabbed a disproportionate amount of attention from virologists in recent years. We weren't granted permission to visit a bat colony in Vietnam, so I went to one in London Zoo instead.
So this is the fruit bat exhibit at London Zoo.
Look at that, yeah. You can see them hanging off the branches. They're very cute little things, aren't
Trying
they?
to find where the fruit is.
So we can see some moving around at the top there.
Yeah, that one's stretching its wings. Look there.
So I'm with Professor Kate Jones. She's a biodiversity scientist at University College London. Kate, you're a bat expert. You're a bat lover, aren't you?
Yes, I am, yes.
And what is it about bats that makes them interesting in the study of pandemics and animal-borne infections?
There's been a recent interest in bats as reservoirs of these pathogens, and it's not clear whether it's because there's more of them in these species or that there's a recent interest in them.
And is the number of viruses that have come from bats disproportionate compared to other animals that we get viruses
Yeah,
from?
that's a really interesting question, and I don't think we empirically really know. Bats are the second most speciose group of mammals, so there are almost 1,200 bat species, so they're going to have a very big variety of pathogens. But people have been showing that bats have got more viruses than rodents, for example.
What kinds of viruses have come from bats in the past?
Yeah, so some really famous ones like rabies, SARS virus and Ebola, but also Nipah Hendra viruses have come from bats.
Nipah and Hendra come from a family of viruses that cause infections of the brain, fatally in more than half of those who catch them. So far, they haven't been shown to transmit from person to person, but many worry that they could gain that ability, something fictionalized in the film Contagion. Real-world outbreaks have killed hundreds of people in Malaysia, Bangladesh and Australia.
The contact rates are increasing rapidly, with deforestation and access roads into the forest and direct interactions like the bushmeat hunting, especially in Africa and Southeast Asia, where people have these big markets of live animals for sale for meat.
This ecological perspective not only explains why there's an increasing risk of zoonotic pandemics, it also has the potential to help us predict where and under what circumstances outbreaks are most likely to happen.
The famous case of Nipah emerging into the human populations is pig farming in Malaysia, where pig farms were intensively developed in rainforests where bats were, and bats were eating the fruit and shedding virus on these fruits, which were then dropping into the pig enclosures, and then the pigs were getting the virus and then spreading it to humans. So indirect and direct contacts increasing all the time as we develop and use more resources.
Preparing for a pandemic isn't only hampered by our need to know more about the networks of transmission between animals and us. The viruses which most worry scientists, the RNA viruses, are continually evolving. They mutate every time they reproduce. And each time, this increases the chance for them to become more virulent or to turn from something that can't transmit between humans to something that can. Mark Woolhouse.
They can change their genes very, very fast. They can do it both by mutation, but also in the case of influenza, by recombination, by mixing up different parts of the genome. So they are able to evolve very quickly, and that's what makes them such a great cause of concern. Some people have calculated that during the course of infection, every single possible mutation is tried out by the virus. And so, yes, this diversity is being generated all the time. Some of that diversity is passed on to the next person or the next animal that got infected, and so the process continues. So the viruses are always exploring the options available to them so they can spread better from and to whatever host they're in.
A big boost to pandemic prediction would be the ability to simply look at the genetic material of a virus and, from this code, work out whether it could infect a person and, more important, go on to spread between people. That would transform our prospects of getting ahead of an outbreak. But getting to this stage of readiness needs some controversial, and some might argue, dangerous laboratory experiments. Are these all your labs?
The whole floor is virology.
Ron Fouchier walking me through his department at the Erasmus Medical Centre in Rotterdam. In the annals of virus research, there's a very famous lab here, one that I'm very keen to see, but Professor Fouchier isn't having any of it.
Well, you cannot go in our BSL-3. We only allow people that have been fully screened, and only people with a real need to know rather than nice to know basis.
Professor Fouchier's team uses the BSL-3 lab, which means Biosafety Level 3, to carry out experiments on the deadly bird flu virus H5N1. The first spread of this virus from birds to humans was detected in 1997. So far there's been very little sign that it can transfer from human to human, but the big fear is that it could. If the virus acquired the ability to live in the upper airways of a person rather than at the bottom of the lungs, that switch would make it much easier to spread by coughs and sneezes. In his lab, Fouchier has transformed the wild H5N1 virus into a version that can spread through the air. The experiments were done in ferrets, the best animal model for influenza in people.
Until we did those experiments, there were many expert virologists around the globe that were of the opinion that H5N1 would never become airborne. And so therefore we investigated whether that was actually true or not. And we came to the conclusion that H5N1 can become airborne, and it has also been shown that an H9 can become airborne, and now with the Chinese outbreak in 2013 of H7N9, it's been shown that H7 can become airborne.
Now explain to me just how complicated it would be for an H5N1 to mutate into something that would be transmissible between people.
We show that only a handful of mutations are enough to make a virus airborne. Now, a handful of mutations for a bird flu virus is nothing. It's close to nothing.
When you say a handful, is there a specific number?
Yeah, 5, 6, something like that. And what we show in these ferrets is once viruses infect mammals, you do see very strong positive selection on mutations that make the virus replicate better in the mammal.
The question is, are the genetic mutations in Fouchier's experiments already out there in the real world? Derek Smith at the University of Cambridge discovered that some were.
We found, of the 5 mutations that were identified, the virus in the wild had, in some instances, many instances, 2 of these 5 mutations, and in a very small number of instances, 3 of the 5.
So, there are many H5N1 viruses out there already in poultry that have 2 of the 5 crucial changes. If these infected a person, how likely would it be that they would acquire the remaining mutations necessary to start spreading between people?
3 mutations is nothing in evolutionary time. It's nothing in a few years or even in a few months. But in the case of a bird flu infection in a human, this is lasting maybe 5 days, maybe 10 days, a couple of weeks at most. And so those 3 mutations would have to happen in a single host, in a single human, within the space of a few days. And so what we did is to set out to answer the question, is 3 a lot?
The work involved computer models of how the virus reproduces and mutates in the human body. These models are being continually refined as new data about the wild viruses comes in.
What we could do, based on the state of the art about what is known of the evolution of these pathogens within humans and transmission, was to say that if the virus had needed 5 mutations, almost certainly that would not happen. It's unlikely that that virus would evolve within a human to be able to transmit to another human. If the virus required 4 mutations, it's pretty unlikely still, but if it only required 3, that actually that is within the realm of possibility, given what we currently know about the biology.
But when you say possible, what does that mean?
That it's the sort of thing that you could imagine happening reasonably. It doesn't mean that it's going to happen tomorrow or next year or the next 5 years, but it's probably not going to be 100 years or 1,000.
The work by Fouchier's group and independent parallel work in an American laboratory upset national security agencies and some scientists, who argued that it was just too dangerous to carry out. But Ron Fouchier says the experiments are crucial in bringing pandemic prediction a step closer.
We can actually start to understand what kind of mutations make a flu virus airborne. And if we can now study how they can make any flu virus airborne, then yes, we already can do better surveillance. We can look in the field whether any of these viruses are more likely to become airborne than others. We can then eradicate viruses from outbreaks, which is not happening right now. But if we would identify viruses in an outbreak in Egypt that already have 3 or 4 of these mutations, I think the advice would be clear-cut, cull the chickens. And so that's a very simple application of the work we do. On the other hand, we rely heavily on vaccines and antiviral drugs if any pandemic will hit. And those drugs and vaccines are currently evaluated with seasonal flu viruses, which have a completely different phenotype from pandemic viruses. Or they are evaluated with true bird flu viruses that do not transmit via the airborne route and therefore are also have a different phenotype. And I think that the best evaluation of vaccines and drugs is using viruses with the right biological properties, those viruses that are virulent and airborne transmitted. And so we now have some viruses that can help us to identify the best drugs and vaccine candidates for future threats.
You're working on flu viruses, but could you use this technique, of course, on other viruses from bats or whatever else that aren't anything to do with flu?
Yes, this is one of the key questions in virology. How do zoonotic animal viruses change to become transmissible in humans? If we can predict that, then we know what to be on the lookout for the next emerging threat. And perhaps even we can come up with intervention strategies.
In theory, we may get to a point where we could find a virus somewhere out in the wild, in an animal or a person, and be able to accurately rate its risk as a pandemic pathogen. But virology isn't the whole story. Do we have the necessary procedures, protocols and cooperation of governments in place to do something about it all? In 2003, China was notoriously secretive about the appearance and transmission of SARS. Jeremy Farrer, head of the Wellcome Trust, says the situation has improved.
It really has changed out of all recognition in that 10 years, and large areas of the response mode is now, I think, reasonable. We've made progress, and you can see that. SARS coming through to H5N1, we had learned a little bit, still huge gaps. Coming through now to H7N9, which is another new virus emerging earlier this year in China, which humans do not have any immunity to. The Chinese response to that, I think, has been exemplary. As soon as it emerged, it was picked up. The information was communicated both privately and publicly to everybody, really, who needed to know about it, openly accessible. They did do a great job.
That said, if a country is reluctant to be fully open and cooperative, there's only so much that international coordinating bodies such as the World Health Organization can do.
There are entrenched policy, political, social reasons why, even though we've moved forward, we have not moved forward far enough. And there are constraints on that. The WHO's great strength is the fact that it represents the member states. That's also, I believe, one of its great weaknesses, because it has to always reach a compromise which everybody can sign up to. So been great progress. We've now got the international health regulations in, so it's actually mandatory that countries report new events. My view is that those international health regulations were in the end a compromise and didn't go as far as anybody, including the WHO, would want in terms of what must be reported.
How could they be strengthened to try and stem some of these problems?
What I would like to see is countries and the WHO working harder together to ensure that we don't just report things, but we put in place a response to those things which is sensible, commensurate with the risk, and doesn't harm individual countries, but actually helps them get through what is an extraordinarily difficult thing to deal with.
There's unease, for example, about what's happening in the Middle East over what's known as Middle Eastern Respiratory Syndrome, or MERS, caused by a SARS-like coronavirus which probably originated in bats. It's killed more than 60 people, most of them in Saudi Arabia. Ron Fouchier argues that the Saudi authorities could be doing much more to assess whether or not this virus poses a pandemic threat.
Well, the key question that was out there from the moment the virus was discovered is how widespread is it already in humans? And the only thing you have to do is go to blood banks or hospitals, take random serum collections from the population and test it against this virus. That can be done in weeks. And the Saudi governments have always argued that there are no validated tests, but you do not need a validated test. There's a very classical test called virus neutralization assay that you can use instantly on any sera, thousands of sera taken from people in Saudi Arabia. If we would know that, we know how widespread it already is. We can do the same with animals. We can just collect blood from camels, pigs, goats, horses at slaughterhouses, and see how widespread the virus really is. That work has not been done. It's so simple to do, and it really provides you with a proper risk assessment of what's going on.
And what would that help you with in terms of what information would that give you?
Well, if this virus is not present at all in Saudi Arabia, we know for sure we're dealing with a very early start of a zoonotic outbreak. If it's already widespread, we're dealing with a disease that's already endemic in the region. If it's a disease that's endemic in the region, we don't need to worry, because that virus has then been around for quite some time and didn't kill all that many people after all. So it's a huge difference in the denominator that you will get of the number of people that are infected with a virus like this. For the animal screening, you would know which are the hosts to be on the lookout for. So if only camels harbor antibodies, then maybe people should approach camels with some care over the next year or so, until this problem is solved in camels.
There are plenty of weak spots in the international infrastructure for virus surveillance, particularly in countries where the outbreaks are most likely to originate. Despite many projects around the world, including Visions in Vietnam, Jeremy Farrar believes we're missing a lot of coverage. And much of the surveillance that is being done is not well coordinated at national and international levels.
In most parts of the world, the veterinary sector is poor. Surveillance in the animal sector is very weak, in some places non-existent. And, of course, there's a difficult balance between illness, trade, the animal sector, farmers, etc. This is a complex dynamic the world over. And so you've got a human sector which has got some major gaps, and you've got an animal sector which is even weaker. And coming back to how I would wish we would go forward, a stronger WHO with greater expertise, greater technical ability and greater investment, but those absolutely same things apply to the animal equivalent to the World Health Organization on the animal side, the FAO.
And when you turn to other kinds of sources of viruses, like wildlife, there really is very little in terms of international regulations that allow people to integrate surveillance or even carry out surveillance of wildlife and integrate it into our understanding of an unfolding pandemic. And that's something that needs attention, getting the wildlife disease surveillance system integrated into the rest of the human and animal disease surveillance systems.
How critical is that, given the next big pandemic will come from animals?
Our first-line defence against anything new, any potential pandemic agent is early detection. That really is the key. And that is going to come about if people working in those different sectors, in the human sector, the livestock sector and the wildlife sector, are talking to each other and communicating about events that they've seen.
At the farm in South Vietnam, Vision's principal investigator Stephen Baker hopes that the integrated and continuous viral surveillance he's leading can be a model for something bigger and even more comprehensive in the future. It's very specific sampling places like this all over the world, actually, that are going to be our eyes and ears to these potential new threats.
Yeah, we're not the only people that are doing this type of study, and there are other projects that are running internationally. They're doing similar kind of things, sampling humans that are coming in contact with a range of different animals. We're hopefully a spoke in the wheel of that type of investigation. So this type of systematic sampling and documentation, we hope, should be a model for the way we actually collect and catalogue and understand the potential of zoonotic transfer at a bigger scale.
The last time the world was on edge about a deadly pandemic was back in 2009, when a strain of swine flu emerged in Mexico and spread within days to the US and Europe. In the end, that outbreak was contained, and the virus turned out not to be as dangerous as everyone feared. But it still caused thousands of deaths. Jeremy Farrar argues that episode shouldn't make us complacent. We urgently need better surveillance to get ahead of the next global killer.
In a cynical world, the events of 2009 may reinforce that sense that scientists are out there to line their own academic careers and the rest of it by crying wolf on a regular basis, and we absolutely have to avoid that. Where you'd like to be is that nobody notices what's happening because you're getting it right, because these events are being picked up early and things are being done to prevent their spread.
If you're being very cynical, someone might argue that you need a massive pandemic to happen for people to sit up and notice.
Yeah, having been through a few of them, I would not wish that on any of us, because it is terrifying.